Okay, so I'll give the second part of my talk. This is unpublished work, so where we have attempted to, address how can we parse the proteolytic and non-proteolytic functions of MMP9 and how MMP9 heterogeneity or functional heterogeneity in MMP9 can benefit or what does it lead to. Okay, so just a quick description about the structure of MMPs in general. There is a signaling domain. Okay, this signaling domain ensures these MMPs are actually secreted towards the membrane. Okay, you have a pro domain. So when the MMP is in an inactive configuration, the pro domain is intact. Okay, this is followed by a catalytic domain. Okay, only when the pro domain is cleaved, then your MMPs are active. And in the back side, this is also equally important. In fact, this is emerging to be of great value because MMPs. So how do you modulate MMP remodeling in a controlled manner, okay, or in a contextual manner, okay, and that is partly achieved through this domain. As you will see, I will kind of speak a lot about these hemopexin repeats focusing on MMP9. These are just some data in various contexts showing how MMP expression correlates with invasion, disease progression. And I draw your attention to this case. So this is for breast and lung. You see, so I have specifically picked 4 MMPs, MMP1, which is a collagenase, MMP2 and 9, both of them are gelatinases. In fact, MMP2 can also activate MMP9, okay? And MMP14 is the one which is transmembrane, as I described in the previous report. Okay, so what you see is MMP2 expression does not change, okay? Its role is more constitutive, and increasing literature are showing that there is a subpopulation of MMPs which reside inside the nucleus, okay? And more so, what we have found that MMP2, MMP9, both of them have an NLS, which means that right after their synthesis, their first destination is actually inside the nucleus and then much later outside, okay? So we are trying to identify what are the conditions which dictate where MMP is localized and in consequence what these MMPs will do, okay? But
so in previous one, looks like MMP1 has a differentially higher expression.
Yeah, so MMP1, yeah, so MMP2 is constitutively expressed. There is no change across cancers, normal or cancer. MMP1 is a collagenase, so it makes sense that in epithelial cancers where there is a lot of deposition of, I think they did not start this meeting, forgot to start. Okay, so that MMP1 is of course involved in collagenase degradation, but it has to be remembered that collagen is a triple helix, right? Its degradation is a multi-step process. So collagen 1 is at action, followed by 9. So they kind of work in tandem. So it's not exclusively MMP1 or exclusively MMP14. Okay. Now, interestingly, there is an increasing body of literature which are showing that MMPs possess non-proteolytic roles as well. So this was one of the first papers from the lab of Mina Bissell at Berkeley. And what this schematic shows is, this is in the context of mammary branching morphogenesis. Okay, when you have these glands which branch, when in dense collagen, so what this molecule is, is your MT1 MMP, which is membrane localized. So for branching morphogenesis in dense matrices, the proteolytic activity is essential, right? However, even in very sparse matrices where pores are already available, what they found was when they eliminated MT1 MMP, its extracellular domain, its catalytic domain, even by deletion, nothing happened. But when they deleted the transmembrane domain, it completely led to loss of morphogenesis, and they subsequently showed that MT1 MMP is involved in stabilizing integrins at the cell surface. So this is nothing to do with degradation, but rather to regulate receptor activation, localization, and its downstream signaling. More recently, MMP9 was also found to, this is in the context of melanoma, where they showed that cells, which, so typically proteolytic cells are well spread, they are, polarized, right? And amoeboidal cells, which migrate in a non-proteolytic manner, are rounded, okay? But to their surprise, so what they found was, in these cases, MMP9 was not necessarily degrading the matrix, but what it was doing was it was binding to CD44, another prominent protein on the cell surface, and stimulating contractility, which means the cell can squeeze through pores. Okay, so again, a non-proteolytic function. Okay, so I have already given you the context of the stiffening in the case of epithelial breast cancer invasion. And what we first asked was in the context of wide number of cancers. So we did it for A549, which is a lung adenocarcinoma, MD-MB231, which is TNBC, as well as HT1080, which is a fibrosarcoma cells. Okay, and what we did was we cultured these cells on something soft or something stiff. So two questions we asked: does the stiffness regulate expression, differential expression, and secretion? Okay, so what you can assess from, so this is the RT-PCR data we have aligned from, even we have taken human dermal fibroblasts, which are normal, MCF10A normal, MCF7 tumorigenic but non-metastatic, and on these three cells which are highly metastatic. So specifically in this group, you see that in MD and HT cells, the fold change in MMP9 expression is nearly 100-fold compared. So on a stiff surface, MMP9 expression is substantially increased. And okay, is the next question is it secreted? So you can collect the conditioned media. So whatever is secreted by the cell, you can collect from the conditioned media, and you can assess, you can perform Western blotting. Okay, so to see that, okay, there is increased secretion also. So increased expression and increased secretion in a stiffness dependent manner. Okay. So we had earlier, this was an earlier study where we showed that when you inhibit MMP proteolytic activity, okay, so you go from soft to stiff, the cells are more migratory, more spread, all of that. Yes, you have a question? Oh, okay. So more invasive. If you inhibit MMP activity using GM6001, which is a broad spectrum MMP inhibitor, the cells are unable to migrate. Okay, and we showed that this defect was associated with integrin levels. So somehow, when you inhibit proteolytic activity, the integrins are displaced, destabilized at the cell membrane. Okay, so we wanted to know how MMP9 is proteolytically as well as non-proteolytically sustaining this phenotype, given that the expression and stiffness dependent secretion of MMP9 was highest, okay, among all the MMPs. Okay, so what you are looking at are just time-lapse images of cells which are on a stiff gel where cells typically migrate a lot in the presence of GM6001, which was the previous case, and now SB3CT, which specifically inhibits only MMP2 and MMP9, not the other MMPs. And you see that SB3CT phenocopies whatever effects we had observed with GM6001, suggesting that these defects are attributed primarily to MMP2 or 9, right? And why does the cell not migrated? So we can perform what are called as adhesion turnover experiments. You transfect with this GFP paxillin, which is recruited to focal adhesions, and you do this imaging to find out what is the average lifetime of a focal adhesion. So if the adhesions are destabilized, then the lifetime will be reduced. Okay, and what we found for across all these 3 cell types, the average lifetime of the focal adhesions was compromised in the presence of inhibitors. Okay. The next set of experiments we did was again to rule out, so between MMP2 and MMP9, who is is regulating it. We did experiments with a MMP2 specific inhibitor, ARP100. Actually, ARP100 inhibits both MMP2 and MMP9, but at low doses, it specifically inhibits MMP2. And now you can see that with ARP100, there is hardly any difference. So we could then pinpoint that, okay, probably MMP9 is one of the prime culprits. Okay, so to assess the role of MMP9 and to parse its proteolytic and non-proteolytic function, what we did was we established stable cell lines where MMP9 was knocked out. It is lentiviral mediated MMP knockdown, and these knockdown cells retransfected either with a full length MMP9 or with a catalytic inactive MMP9. Right? So the only difference is the catalytic inactive MMP9 is incapable of degrading the matrix. Okay. Now, what you can clearly see that while knockdown cells rescued the cell spreading, knockdown cells led to reduction in cell spreading, re-expression of either full length MMP9 or the catalytic inactive mutant, both of them did the rescue, suggesting that for spreading, my catalytic activity is not essential. Okay. So we did the same set of experiments then, okay, what so in the context of migration. Okay. We did the same experiments, and once more we found that delcat, which is the inactive MMP9 re-expressed cells, they rescue motility to the same level as full length MMP9 cells, which means that for migration also MMP9 catalytic activity is not essential. However, when we entrapped these cells, okay, when we entrapped these cells in 3D collagen matrices and tracked their migration pattern, what you can clearly see is knockdown cells unable to invade, full length cells they have rescued, but the catalytic inactive mutant is not able to rescue 3D invasion. So this suggests that while the spread morphology and 2D migration can be sustained by MMP9 independent of its catalytic activity, catalytic activity is absolutely essential when the cell is migrating in a 3D matrix filled with collagen. Okay, so how is MMP9 doing this? Okay, so to address that question, what we once again looked at was the formation and dynamics of focal adhesions. And what we found was, knockdown cells had lot less number of focal adhesions, and the focal adhesions were unstable, they had limited lifetime. But this was rescued by my delcat variant, suggesting that MMP9, and you see that in the integrin expression as well. So suggesting that MMP9 is capable of rescuing or sustaining the cell spread phenotype through regulation of integrin adhesions. Okay, so how does MMP9 interact with integrins? So we dug up, so again what I'm showing you is the entire structure of MMP9, and so once again you have, this is your catalytic domain, pro domain signaling peptide, and there is your hemopexid repeats. Okay, and what has been reported in the literature in the context of melanoma cells was that this hemopexin domain 4 has a sequence of amino acids which are responsible for binding to integrins. Okay. In addition, right, the catalytic domain, in fact the site right next to the catalytic site there are some fibronectin repeats, and specifically most of you have heard of RGD peptide, right, which is used for adhesion. There is actually an RGD sequence present in the catalytic domain. Okay. So we thought that probably it is via one of these two sites that MMP9 is somehow stabilizing integrins at the cell membrane. So to answer that question, we established the following mutants. We deleted the 17 amino acid sequence. So this we are referring as the delta beta 1 sequence, or we mutated this RGD site to RGE. So you can have a delcat, which we had already established before. There is no proteolysis, association is expected. The RG mutant, ideally proteolysis is unperturbed in these cells, right? Delta beta 1 proteolysis should be unperturbed, and delta beta 1 delta cat, where in the inactive mutant we have also removed this del beta 1 site. Okay, so there should be no proteolysis and presumably no association. So we asked first whether there is any association between MMP9 and integrin, which we can specially localize, and where do these associations occur between MMP9 as well as in the different mutants. Okay, so this we did using an assay called proximity ligation assay, which can detect proteins in close proximity. Okay, so what you are looking at is, so all these mutants express the MMP9. This is the dual link is the signal that you are detecting. So if they are very proximal, then you have rolling cycle of amplification, DNA amplification. When you add these fluorescent probes, those sites are detected and it gives you green fluorescence. Okay, so what you can see is in, let's say, full length and delcat, means catalytically inactive set, at the periphery you have, where you have MMP9 localized, you also have the presence of these green dots. In RG, surprisingly, this is still there. So when we converted from RG to RE, it's still there. But when we removed del beta 1, so when we made this deletion, the green signal is significantly reduced. So this is the intensity showing that in full length delcat and RG cells, you have association, right? And this association, so in the center, of course, there's a blob. It's difficult to say, but at the peripherally, in these cases, you see prominent association, which is substantially reduced in both del beta 1 and del beta 1 delcat cells. So when you remove the 17 amino acid integrin binding site, this association is lost. But in RGE, where this 17 amino acid residue is there, but you have mutated the RGD to RG, you can still detect association at the cell. So we then asked, okay, what is the functional relevance of this perturbation? And once again, in the top we are showing the 2D migration, and in the bottom we are doing the 3D invasion experiments. So what you see is, in this case, again with RG, so even though there was association between MMP9 and integrin detected in RG cells, but 2D motility was impacted. Same is true in del beta 1 or del beta 1 delcat cells. Okay? So there is this difference that in our dual link signal, which suggests that yes, integrin can associate with MMP9, but in your RGE cells, but they are unable to migrate. And of course, in 3D invasion, all these 4 mutants are unable to sustain invasiveness of these cells. But if we, in these cells, if we add externally, we supplement with conditioned media secreted from full length MMP9 expressing cells, we can rescue the phenotype. Okay? So how can we understand the difference between the RG and the delta beta 1 results? Okay, so what we hypothesized was one possibility that the RGD to RG mutation is somehow impacting integrin stability at the cell membrane. Okay, so which we functionally assessed using an AFM experimental setup, where we functionalized our AFM probe with RGD. We bring the tip bring it in contact with the cell surface. We give a time delay. So if there are some integrin molecules which are there on the cell surface, they should bind to the RGD. And when I retract my tip, these bonds of adhesion should break, giving rise to an unbinding force peak. So the maximum force here corresponds to the maximum number of bonds that were formed between RGD and integrin on the cell surface. Okay, so I can quantify that force, and what you find is in full length and delcat cells it is comparable. In RGE cells it is dropped similar to delta beta 1 and delta beta 1. So suggesting that this interaction is somehow controlling how many integrin molecules are actually reaching the membrane, or how long they are stable on the cell membrane, so that they can form adhesions. So how do we assess that question? So we did some tricolor experiments wherein MMP9 and integrin beta 1, both of them were tagged, and we also labeled with biotracker. So biotracker will label all vesicles inside the cell, right? So we track their, let me replay the video. Okay, so you see the MMP9 molecules going to the periphery, integrin molecules also following, and same is true for the biotracker signal, right? Which suggests that this association is essential for their transport to the membrane. So somehow they are being co-transported in the same vesicle. Okay, co-transported in the same vesicle. So I can quantify the speed of these integrin foci which are reaching the membrane, and what you see is for full length, delcat and RGE, in all these cases the foci speed are comparable, which means that in all these cases that MMP9 and integrin are actually going to the periphery of the cell. However, so here you see another difference between RG and delta beta 1. When you remove that 17 amino acid stretch, integrins cannot reach the membrane. So delta beta 1 or delta beta 1, both of them, the foci speed is very low. So they are mostly restricted to in and around the ER and not co-transported. Okay. What about MMP9? Is there any importance of this interaction so far as MMP9 secretion is concerned? To assess that, what we did was we collected the conditioned media from these cells, okay, and we probed that is MMP9 present in the conditioned media. What you can see in full length, you have, and this is, so basically the top band is gelatin zymogram. You can see high activity of MMP9. In delcat, of course, expected there is no activity, made it non-catalytic. In RG, there is some, you can detect, but it is reduced partly. But in the other two cases, you can see very weak, and in total MMP9 also you see that there is no secretion, suggesting that this association is not just transporting integrins to the membrane, but is also essential for MMP9s to be secreted. Okay. Now comes the question that in what form is MMP secreted, right? In what form? So from the previous talk, I tried to say that, okay, there must be a mechanism that the local density of MMPs are enriched, okay? And we hypothesize that one of the ways in which the MMP9 might be secreted is in the form of exosomes, okay? If you look at the exosome biogenesis pathway, these are vesicles from which you have intraluminal vesicles which bud off from the surface inward, okay? So when the outer membrane fuses with the plasma membrane, the inner vesicles which have bud off, they are secreted, and these are exosomes, okay? Roughly 100-200 nanometer in size. Okay? So we isolated the exosomal pellet and we probed whether MMP9 is indeed So first of all, whether there is increase in exosome number, what you can clearly appreciate is increase in stiffness leads to significant increase in the secretion of exosomes, and by doing gelatin degradation experiments where we incubate fluorescently labeled gelatin coats with my exosomal pellet, we can detect these degradation spots. So mind you, these degradation spots are in the length scale of cells, not single molecules which we cannot detect. So there must be a mechanism for exosomes also to maybe aggregate and sediment, thereby creating such big patches. Okay, and we did these experiments of adhesion wherein we seeded cells in the presence of exosomes. Okay, and we see that, so if you just plate these cells for half an hour and then quantify how many cells have actually attached and what is the average area of the cells, you see the cells which have been supplemented with 5 kPa exosomes, they are maximally attached and they spread the most. And you can look at phosphofax signaling to show that exosomes are actually modulating this adhesion signaling. Okay, so based on this, we propose a model wherein MMP9 via its 17 amino residue in the hemopexin domain leads to its association with integrin, and both of them are trafficked to the cell membrane. At the cell membrane, we propose, since RG is leading to destabilization of integrins, we propose that the integrins are held in an active configuration by the RGD site of MMP9, and the exosomes which are secreted, they are mediating the degradation, and there's a switch from integrins attaching to MMP9 to integrins actually attaching to the matrix. Okay. Now, okay, so far so good. So we think that all cells should migrate equally, right? Now think of a population level behavior. You look at the MMP9 heterogeneity, our default expectation is all cells have trunk loads of MMP9, right? But what this plot clearly shows is you have only a small fraction of cells which have high MMP9. Rest of them are very low. So what can be the implications of such heterogeneity at the population level was the question that we asked. And for doing that, okay, so once again, we took these cells that we have established before, right? My knockdown cells or SHMMP9 cells, same thing, and cells re-expressing the catalytic inactive mutant. Okay, so what you can see is when we knock down the cells become very small as well as very soft, and we can recover the cell properties when we re-express this catalytic inactive mutant, suggesting that the cell biophysical properties which I tried to convey are an important indicator of invasiveness are also tuned depending on the MMP9 activity. Okay, so how do we address this question? So we again made use of a cellular POTS modeling framework, but now what we did was we have a group of cell cluster comparing variable proportion of cells which secrete MMP and proportion of non-proteolytic cells, which are, so they are depicted as smaller in size because this is what experimentally we observed. Okay, so I can do these simulations, vary the proportion of proteolytic and non-proteolytic cells, and what clearly, of course, the expectation is that increase in the proteolytic fraction would lead to increase in the extent of invasion. Okay, we can quantify this data here. Okay, that as you increase the proportion of proteolytic cells, the overall displacement or in extent of invasion increases, but note that this is not linearly proportional. So there is some, you can, if you follow this track, beyond 60% it begins to saturate. Okay, so this is not a linear correlation. Okay, and if I parse into the little bit detail that for each of these combination, how much did the proteolytic cells migrate in relation to how much the non-proteolytic cells migrated. And what you can see from this plot is, okay, there is no difference or hardly any difference. Okay, even where you detect statistical difference, it is very low. So even though proteolytic cells are capable of degrading more matrix, there is no difference between theirs and non-proteolytic cells. Okay, so since we do these simulations, we have from each simulation we can ask, okay, what were the spatial parameters which might impact this observation. Okay, so what we did, because we had the displacement information calculated from simulation, we also know what is the measure of circularity, right? Whether it is rounded or whether it is stretched out, that we can find out how, because we have information about the area as well as about its perimeter. I can find its circularity. I also know where this cell was initially positioned in the cell cluster, which means that if you had a cell which was at the periphery, the expectation is this cell will by default migrate more than if the cell was positioned deep interior. And secondly, we hypothesized that, okay, for the non-proteolytic cells, maybe they are hitchhiking a ride with the proteolytic cells. In other words, if they have greater number of proteolytic neighbors, they stand a greater chance that they will actually invade. Okay, so with these parameters, we did a simple TSNE mapping, and then we specifically looked at the cells, okay, within the non-proteolytic fraction which migrated more or exhibited more displacement. So I've highlighted those particular cells. Okay, what you clearly see is these cells which had invaded more, number one, had either Ri was greater, which means they're already at the periphery to begin with, or in some cases they were next to proteolytic cells, so they were hitchhiking a ride. Okay, but there was another observation which we found was independent of the localization of proteolytic or non-proteolytic cells. On an average, you see that the non-proteolytic cells possess a lower circularity. What does it mean? That they are more deformed. Okay, so this is quantified here. I can, because I know how the circularity evolved for each cell, I can find its average plot of circularity, and you can clearly appreciate that the non-proteolytic cells possess a lower circularity compared to the proteolytic cells. This is particularly evident at high cell-cell addition where JCC is low. Here you see the difference reduces because the proteolytic cells are scattering. There is no cell-cell addition. Okay? And we also did whether there was any particular correlation between displacement and circularity. But what you see is these are pretty uniform distributions. There is no particular correlation with displacement. So irrespective of where these cells were present, they are displaced substantially. Okay, so this is again, the same information plotted as a function of what were the initial position. Okay, so consistently the same thing stands out, that the non-proteolytic cells are possessing lower circularity, which means that they are getting more deformed. Okay, so based on that, we thought, okay, maybe the difference in physical properties of proteolytic and non-proteolytic cells is causing the proteolytic cells to squeeze past the non-proteolytic cells, okay, if they were deep inside the cluster. Okay, so before we did that, what we also did was, since for a given simulation realization, okay, we already know what is the position of each proteolytic and each non-proteolytic cell, I can do a correlation between their direction vectors. So if they were closely following each other, then my cos theta should be close to 1, and the average distance of separation between those two cells should not increase beyond a point. Okay, and which is what we saw, right? So the highly motile non-proteolytic cells, you see the value of cos theta is always harboring close to 1, which means that the non-proteolytic cells are very closely following the proteolytic cells, and the Dij is always within a limit. And even during this exercise, you see that the non-proteolytic cells which are in back, they are getting deformed to a greater extent, right? As a function of time, their circularity values keep dropping. Okay, so which means that maybe the proteolytic cells are coming to the fore. In contrast, we can also look at cells where a proteolytic cell and a non-proteolytic cell maybe were initially closely placed, but as a function of time, you see that the distance kept on increasing and cos theta values, there is no correlation, suggesting that in these instances, the non-proteolytic cells were not really following the proteolytic cells. They are just kind of, maybe following some other proteolytic cells. So how do we test the hypothesis that my proteolytic cells are somehow squeezing the non-proteolytic cells? That is one possibility. And or the non-proteolytic cells, by virtue of becoming or being much smaller than the proteolytic cells, are increasing the entropy of the system, giving more dynamics possibility. Okay, so we did these following simulations. So this was the original. So take a close look at the way I have drawn the proteolytic cell and the non-proteolytic cell, right? This is the true picture. If I knock down MMP9, cell becomes small, and this rugged cell morphology is supposed to indicate that it is more deformable, wobbly. Okay, I can have a situation where the non-proteolytic cell is smaller in size, but of identical stiffness to that of the proteolytic cells, or both of them are same in size but vary in deformability, or the other extreme which we have with our Delcat cells, that not only are they same in size, they are also same in deformability. And I tracked the translocation for each of these conditions, and you can clearly appreciate, irrespective of the proportion of proteolytic cells, you have the maximum drop with the blue condition. So which means that it is basically the combination of softness as well as their small size which is enabling the proteolytic cells to come out, thereby driving invasion of the collective population. Okay. So to test this, what we did was we made these spheroids either using control cells, right? Control spheroids, knockdown spheroids, non-proteolytic, so these are my Delcat cells, or I combine 1:1 ratio control and knockdown cells, or I combine control and non-proteolytic cells. Okay. In either instance, so of course you see that there is a variation in their spheroid size. It's not that they are equally compact. So in all probability, the extent of E-cadherin expression has also changed. Even though these cells have very low E-cadherin, still you see that the knockdown cells tend to be more compact than the control cells. So to look at what is the impact on invasion, what we did was we implanted these spheroids in 3D collagen matrices and we looked at their invasion. However, given that the spheroid sizes were not same, what I did was the day 0, you see that we have divided the zones into 3 zones. My zone 0 is the initial, at the day when I seeded the spheroid, it is the circle encompassing the spheroid. And then you have dotted lines which are of equal radial width. Okay, so I want to disregard the possibility that of size differences in my overall scheme of things, right? And I look at day 1 or day 2, and I look at, okay, so at day 1, what are the proportion of cells, okay, in each of the zones. Okay, so and I not, because this number of cells is a random thing, right? From experiment to experiment, you cannot compare. So we normalize the total number to 1 and then identify what are the zone wise distribution. Okay, so what you can clearly see is even at day 1 or day 2, knockdown and non-proteolytic, none of them are able to invade. But the most interesting observation is here. When I combined the control and the knockdown cells in 1:1 ratio, I get the maximum amount of invasiveness. Okay, this is even more than my control. So suggesting that proteolytic heterogeneity is actually benefiting me in some ways. Okay, And the same difference is amplified at day 2. Okay. When I combine my control and non-proteolytic spheroids, they are not migrating as much. They're just similar to my control cells. Okay. And these are just like a sequence of images that I took from high resolution imaging to show. So this is an example where a knockdown cell is getting, so you have the emergence of a green proteolytic cell. It is stretching out and it is basically bypassing this this knockdown cell to come forth. Okay, so that is how, by virtue, and you can also appreciate the extent to which the non-proteolytic cell is getting squeezed out. Okay, so based on this, we propose this model wherein by tuning deformability and cell size, okay, so there is this intimate crosstalk, right? So more MMP secretion, the phenotypes are more spread out, but there's these cells also more stiff or less deformable, and lesser expressing MMP9 are much more deformable and tend to be softer. So you can think of this scenario where a proteolytically heterogeneous mixture of cells will lead for the heavily proteolytic cells, even though they are they may be internalized in the cluster, they can push out the non-proteolytic cells, come out at the periphery and serve as leader cells. Okay, so though I did not harp on this, another aspect of this work is that the energy cost of a cell to maintain its proteolytic state is much more than a non-proteolytic cell. So even, which means that even in the context of constrained energy, either you talk of hypoxia or you talk of nutrients, you would see the same situation turn in where there would be a greater proportion of non-proteolytic cells. I think that's all I have. So I have these students who have done the work. So Abhishek, Sandeep were from the previous first talk, and Sarvajit Asad are from the current talk. Okay, thank you.
Questions?
Which biotracker? Oh, okay, biotracker. It is, so basically it will label all vesicles. So it has a fluorescent tag, and it will label all vesicles within your cell. No. It is a real entity. So you have MMP9 and integrin, which are labeled with 2 colors. Now, how do you know whether they're being co-transported? Okay, so biotracker is that label which will specifically label vesicles. So then you can colocalize your integrin and MMP9 to the same vesicle. Yeah. Impact of microgravity, it's anybody's guess, I have personally not come across any such paper. Okay. So when your the gravity conditions are altered. Okay. You can stop.
Okay, if there are no other questions, let's thank Shamik once again.